Good morning, students. So, in the last session, we talked about DNA replication in eukaryotes. Today, we talk about DNA replication in prokaryotes. We multicellular organisms are called eukaryotes. We have a linear DNA and it replicates in the S phase of interphase by a process called as semi-conservative method of replication. Bacterial DNA, unicellular cell DNA is a circular DNA. Unlike our DNA, which is linear, bacterial DNA is circular. It replicates by a mechanism again called as semi-conservative method of replication. So both eukaryotic and prokaryotic is semi-conservative, but our DNA is linear like a line. Bacterial DNA is circular. And let us see what is the difference between eukaryotic and prokaryotic DNA replication. For that, first, let us understand कि bacterial DNA दिखता कैसा है. In prokaryotes such as E. coli, they do not have a well-defined nucleus. The DNA is not scattered in the nucleus. Bacterial DNA, ये है bacterial cell wall with the cell membrane. And this inside is the nuclear DNA. There is no nuclear membrane. So nuclear membrane नहीं होता है. Unlike we have cell membrane and nuclear membrane, bacterial DNA has cell membrane but no nuclear membrane. So there is no nucleoplasm. There is only cytoplasm. And in the cytoplasm, the DNA is present. The DNA is held with some proteins in a region called nucleoid. And the DNA in the nucleoid is organized into large loops held by proteins. So this is the cell membrane. This is DNA attached to it. DNA is in the form of coils. DNA coil form में होता है. So these are the coils of the DNA, and these DNA coils are held together by a protein. And proteins will hold the DNA together. Of course, these proteins are non-histone proteins. So, bacterial DNA के अंदर DNA होता है proteins के साथ, but our DNA is DNA wrapped around histone proteins. Bacteria has coils of DNA held together by non-histone proteins. Non-histone proteins. And so, if the length of the E. coli DNA is 1.36 millimeter, can we calculate the number of base pairs in the E. coli? If you remember, one base pair is 3.4 angstrom units or 0.34 nanometers. And how many base pairs are there in the DNA? We had done that in the lecture before. E. coli has 4.6 into 10 raised to 6 base pair, 4.6 into 10 raised to 6 base pair multiplied by 0.34 nanometer turns out to be 1.36 millimeter. If you remember, our DNA was 2.2 meters. Bacterial DNA is 1.36 millimeter, 4.6 into 10 raised to 6 base pair multiplied by 0.34 nanometer for distance between two base pair turns out to be a total of 1.36 millimeter. And this is the DNA in the interface. Sorry, in the interface. When this DNA goes into replication phase, DNA जब replication में जाता है, all its proteins disappear and the DNA looks like a circular DNA. So this is the circular DNA, and it has two coils held together to the cell membrane. And this is DNA during replication. So let us see how replication actually takes place. So this is replication in case of bacteria. Prokaryotic DNA, there is a circular DNA present. यह है circular DNA, and there is only one origin. So origin of replication एक ही होता है, and that replication is called theta replication. So the bacterial DNA replication will look like theta and is called as theta replication. So this looks like theta and is called as theta replication. What do you mean by this? इसका मतलब क्या है? Let's see how bacterial DNA replicates. This is the bacterial DNA made up of two DNA strands. Two DNA strands spirally twisted around each other. Circular DNA. There is only one origin of replication. Unlike human DNA, where we had multiple origins of replication, bacterial DNA में सिर्फ एक origin of replication होता है. At our multiple sites of DNA, origin of replication, an incision is made with the help of incision enzyme, endonuclease, and will cut our DNA into multiple fragments. In case of bacteria, at the origin of replication, there is no incision, no incision, no endonuclease will cut the bacterial DNA. अगर इंसिशन एंजाइम एंडोन्यूक्लियस ने कट किया, बैक्टीरियल डीएनए इंस्टेड ऑफ सर्कुलर विल बिकम लीनियर. तो सर्कुलर डीएनए लीनियर हो जाएगा. वी डोंट वांट दैट. So at the origin of replication, there is no incision made by endonuclease. So what will happen at origin of replication? So here we have adenine pairs with thymine, guanine pairs with cytosine, cytosine pairs with guanine, thymine pairs with adenine. At the origin of replication, the enzyme called helicase will start breaking hydrogen bonds in both directions. So helicase will break hydrogen bonds in both directions, and the two DNA strands will start separating. Both DNA strands start separating with the help of helicase. And at one point, this is the origin, this is the origin, this is the new strand. Sorry, the old strand template. This is the old strand. This is the old strand. This is the old strand. And यहाँ separate नहीं हुआ है. So we have the old strands. So this looks like the Greek alphabet theta and is called as theta replication. इसको समझने के लिए समझो I have a circular sticker. Say you have just become a doctor and your papa has gifted you a car. So you want to proudly put your doctor sticker on your car. So you take a sticker, उसके ऊपर प्लस साइन है, एंड उसके पीछे होता है पेपर. तो you catch hold of the sticker with the paper on it, and you catch hold of the sticker and the paper and start removing it. The sticker starts coming out of the paper, and removal will take place clockwise as well as anti-clockwise. And जब आधा स्टिकर पेपर से सेपरेट हो गया है, तो यहां है पेपर, यहां है पेपर, पेपर के ऊपर स्टिकर, और स्टिकर अब आधा निकला हुआ है. सो दिस इज योर पेपर, दिस इज योर स्टिकर, और यहां दोनों स्टिकर और पेपर है. सो this is looking like the Greek alphabet theta and is called as theta replication. Theta replication. And check this carefully. This is the old strand. This is the old strand looking like the Greek alphabet theta, and from the old strand, origin of replication, DNA polymerase. Which enzyme, sir? DNA polymerase will start supplying new nucleotides. DNA polymerase will supply new nucleotides. DNA polymerase will supply new nucleotides. So, old strand को मिलेगा, ये है old strand, and old strand को मिलेंगे new nucleotides. ओल्ड स्ट्रैंड को मिलेंगे न्यू न्यूक्लियोटाइड्स. सो व्हाट आर वी हैविंग? फॉर्मेशन ऑफ अ डॉटर डीएनए where there is old strand with new nucleotides happening bidirectionally, and even in this strand, this is your old strand, and old strand के ऊपर bidirectionally you have new nucleotides. Bidirectionally you have new nucleotides. So old strand has new nucleotides coming bidirectionally, and this is your theta. Now what will happen? सर, ये आपके सिलेबस में दिया है, बट एक बहुत ब्यूटिफुल पॉइंट जो आपके सिलेबस में नहीं दिया है. सर, इफ दिस इज योर सर्कुलर डीएनए, इसको जरा ध्यान से देखना, एंड इट हैज गॉट 100 कॉइल्स. इसमें 100 कॉइल्स है. नाउ I have origin of replication, and at the origin of replication I start forming the origin of replication पे you have new bubble, एक bubble बनने लगता है. Yeah, this I'll just show you properly. So this is your circular DNA, and at the circular DNA you have origin of replication where it starts separating. And as it separates, ये हो गया एक replication bubble. Check this carefully. Replication bubble. तो replication bubble है, and how many coils do we have? If you remember, we had 100 coils. Have we made incision? No, no incision. So when the replication bubble ये जो बनने लगा है, this is replication bubble. यहां कितने coils आ गए? 50. और यहां कितने coils आ गए? 50. So original coils कितने थे? 100. और यहां कितने coils है अभी? यहां कितने coils है? 50. और यहां कितने coils हो गए? 50. Number of coils will remain the same because we have not made incision. So coils will remain coils, and at one point in time, as the replication bubble becomes bigger and bigger, the coils come very close to each other. Check here. यहाँ coils में distance ज्यादा है, but जैसे ही replication bubble बड़ा बनता है, coils are coming close to each other. And as the coils become close to each other, there develops a rotational pressure, which is called as torque pressure. तो rotational pressure या torque pressure आता है. Let us see that diagrammatically. So, if we see this carefully, ये है strands और ये है strand, और इनके बीच में बनता है a bubble called as replication bubble. And यहां पर coils come very close to each other. Coils come very close to each other. Coils come very close to each other. And ये पार्ट में नॉर्मल कॉइल है, ये पार्ट में नॉर्मल कॉइल है. बट जैसे ही रेप्लिकेशन बबल बड़ा होता है, the coils come very close to each other, and in this highly coiled area, there develops a rotational pressure, which is called as torque, torque pressure. And because of torque pressure, कॉइल्स में इतना टॉर्क प्रेशर आ गया है कि रेप्लिकेशन बबल अब बड़ा हो ही नहीं सकता है, which means the two strands in the theta cannot separate because of this rotational pressure or torque pressure. So you have to relieve the torque pressure. And how is the torque pressure relieved? The torque pressure is relieved by an enzyme called as topoisomerase. Topoisomerase or DNA gyrase. Topoisomerase or DNA gyrase, ये है torque pressure, very close to each other. And topoisomerase will cut one of the DNA strands. तो DNA strands को cut कर देगा. ये है रोटेशनल प्रेशर, और यहां पर एक कट आ जाएगा, इंसिशन आ जाएगा. एंड बिकॉज ऑफ द इंसिशन द डीएनए स्ट्रैंड विल अनकॉइल. तो समझो ये पिक्चराइज करना है, ये है नॉर्मल डीएनए, और बिकॉज ऑफ रेप्लिकेशन बबल बिकमिंग बिगर, साइड्स में क्या आ जाएगा? साइड्स में आ जाएगा रोटेशनल प्रेशर और टॉर्क प्रेशर. तो यहाँ कॉइल्स बहुत ज्यादा कॉइल 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 हो गया है, कॉइल 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 हो गया है, इसमें रोटेशनल प्रेशर या टॉर्क प्रेशर आ जाएगा. तो एंजाइम कॉल्ड डीएनए गायरेज और टोपोआइसोमरेज आएगा, एक स्ट्रैंड को कट करेगा. एक स्ट्रैंड को कट करेगा तो जितना भी रोटेशनल प्रेशर हुआ था, अब जैसे ये कट हो गया है, दैट स्ट्रैंड विल अनकॉइल. तो वो अनकॉइल हो जाएगा. यहां पर दोनों स्ट्रैंड्स कॉइल्ड है, एक स्ट्रैंड को कट किया, कट एंड में immediately the DNA strands will uncoil and the torque pressure is relieved. And after the torque pressure is relieved, the two cut ends will stick again. The two cut ends will stick again by the same enzyme called as DNA gyrase or topoisomerase. So, ये है lot of rotational pressure. Incision is made here. Now, this strand is here, but the other strand will start moving in the opposite direction. Opposite direction में strand starts rotating in the opposite direction and release the torque pressure. Torque pressure relieve हो जाएगा, and then the cut ends are stuck to each other, and the DNA again becomes coiled. So ये coil था, highly coiled, यहाँ cut हुआ, and torque pressure relieve हो गया, and the cut ends again stick. So this will again look like this. तो ये वापस से ऐसे दिखेगा. तो ये पार्ट पहले कैसा दिख रहा था? Like this. It was torque pressure, lot of torque pressure, lot of torque pressure. And once the torque pressure is relieved, it will look like this. तो समझो मैं यहां दिखा रहा हूं, ये है torque pressure. This is with torque pressure, with torque pressure. And once the DNA gyrase or topoisomerase cuts it and relieves the torque pressure, now it will look like this. So this is without torque pressure. This is without torque pressure. And that way torque pressure निकल चुका है. This was with torque pressure. वो रिलीव हो गया है. दिस इज विदाउट टॉर्क प्रेशर बिकॉज ऑफ डीएनए gyrase or topoisomerase, and then helicase will continue breaking hydrogen bonds. Helicase will continue breaking hydrogen bonds, and the two DNA strands will completely separate from each other. Helicase breaks hydrogen bond, तो एक strand यहां आ गया और एक strand यहां आ गया. तो जैसे हमने वो पेपर और स्टिकर में से पेपर और स्टिकर सेपरेट हो रहा है. आपकी उंगलियां कौन है जो पेपर और स्टिकर को सेपरेट कर रहे हैं? पेपर और स्टिकर को सेपरेट कर रहे हैं आपकी उंगलियां कौन है? डीएनए हेलिकेज एंजाइम. इट इज ब्रेकिंग द हाइड्रोजन बॉन्ड बिटवीन नाइट्रोजन बेसेस. तो पहले ऐसा था, फिर वो ग्रीक वर्ड थीटा दिख रहा है. एंड एज फर्दर हाइड्रोजन बॉन्ड्स ब्रेक, two DNA strands completely separate. So we got old strand completely. And at the origin of replication, helicase broke the hydrogen bond. And DNA polymerase starts supplying new nitrogen bases clockwise, new nitrogen bases anti-clockwise. DNA polymerase supplies new nitrogen bases clockwise, new nitrogen bases anti-clockwise. And जैसे ये सेपरेट होगा, यहां पर सेपरेट किसने किया? हेलिकेज ने. और न्यू स्ट्रैंड क्लॉकवाइज और एंटी क्लॉकवाइज किसने बनाया? डीएनए पॉलीमरेज ने. सेपरेट किसने किया? हेलिकेज ने. और न्यू न्यूक्लियोटाइड्स क्लॉकवाइज और एंटी क्लॉकवाइज किसने सप्लाई किया? डीएनए पॉलीमरेज ने. तो वन मदर डीएनए will become two daughter DNAs, first looking like the Greek word theta, and eventually theta separates out to form old strand. ये हो गया old strand, old strand. और उसके ऊपर ये आ गया, डॉटेड में दिखा रहा हूं, न्यू स्ट्रैंड. डॉटेड इज द न्यू स्ट्रैंड. डॉटेड इज द न्यू स्ट्रैंड. So, what are we having? The bold is the old strand and dotted is the new strand. So, we are again having one mother DNA forming theta, and eventually two daughter DNAs, each having one old strand and one new strand. So this is also called as semi-conservative method of DNA replication. Semi-conservative method of DNA replication. So we ended with it. What is the difference between eukaryotic and prokaryotic DNA replication? In eukaryotic DNA replication, there were multiple origins. In prokaryotic, there is only one origin. In eukaryotic, origin was broken by endonuclease and बहुत सारे fragments बन गए थे. Here we have only one origin and no incision is made by endonuclease. So circular DNA remains circular. And what is broken on either side? Hydrogen bonds by helicase, which then looks like the Greek alphabet theta. Theta, and it's called theta replication. The torque pressure is relieved by topoisomerase or DNA gyrase, which will remove the torque pressure. And new nucleotides are supplied by DNA polymerase. So याद रखना, there was no endonuclease. Hydrogen bonds were broken by helicase. New nucleotides were supplied by DNA polymerase, and one mother DNA became two daughter DNAs one original strand, one new strand, one original strand, one new strand. So this is also the semi-conservative method of DNA replication. चलो ये याद रखना, बायोटेक्नोलॉजी चैप्टर में ये प्रोकैरियोटिक डीएनए रेप्लिकेशन आपको बहुत काम आने वाला है. सो रिमेंबर इट फॉर द चैप्टर ऑफ बायोटेक्नोलॉजी. चलो, आई होप यू अंडरस्टूड दिस. टेक केयर.